Good morning, everyone. It's my task this morning to share with you the accomplishments of the Drug Development Coalition. In a way, this is a bit of the name for this group is a bit of a misnomer, and I'm going to divide my talk into two portions. A short portion talking about the structure of the Drug Development Coalition, and then the majority of the talk talking about the accomplishments of CPTR and actually others outside of CPTR in drug development over this past year. Deborah showed you this slide of the organizational structure of CPTR. It's divided into three arms: the Regulatory Science Consortium, the Research Resources Group, and the Drug Development Coalition. Now, the Drug Development Coalition is open only to those organizations that have drugs in clinical development. So many of you are not part of this coalition, and I know in the past there's been a sense that there's something mysterious going on here. So I want to just be very clear on what this coalition is. It's a legal entity really put in place to allow companies to come together and discuss their clinical data in a pre-competitive environment to protect them from antitrust issues. And then companies that come together in this framework are allowed to make agreements with each other, go off and do collaborations. So this is really a legal entity, and I'll show you the signatories of this entity. It's AstraZeneca, Bayer, Johnson & Johnson, Sanofi, TB Alliance, and the TDR arm of WHO. So these organizations have felt that, that they can avail themselves of this entity to talk to each other. I encourage any other organizations who will find this useful to help them talk to each other to sign up and join. But I think for the purposes of this meeting, what I want to do is talk more broadly about what's been accomplished in drug development over the past year, and I think we've had some pretty significant accomplishments. The global TB drug pipeline is shown here. I'm not going to go through this. The Working Group on New Drugs keeps this up to date. The bottom line from this message is that we are in great shape compared to where we were 10 years ago. But since we all know the attrition rates in drug development, we need to get into better shape, and that's why we're all here. To share with you some of the key recent events over the last year. You're going to hear more detail about these tomorrow from each of the sponsors. The TBTC studies looking at rifapentine and INH given once weekly for 3 months in latent tuberculosis were very successful and resulted in the CDC recommendation in December of last year to use these regimens in the U.S. Otsuka has filed for approval of delaminid in MDR-TB in the European Union, and Janssen has filed for approval of bedaquiline in MDR-TB in the U.S. and Europe. Hopefully by next year we will no longer be able to say there's been no new drug approved for tuberculosis in the last 50 years. I want to share with you some key findings that were presented at the TB Clinical Pharmacology Workshop a couple weeks ago, a satellite of the ICAC meeting, because I think these are very significant. The first is from the Panacea organization. That's a clinical trials consortium that's funded by EDCTP in Europe. Martin Barret presented his data on high-dose rifampicin. The idea here is to take the dose of rifampicin higher than the standard dose of 10 milligrams per kilogram, indeed take it as high as possible, and then ask whether a higher dose of rifampicin inserted into the standard treatment regimen would result in treatment shortening. This is exciting because it would be an easy win if it works. Martin presented data showing that in a 2-week study they've gone up now to 35 milligrams per kilogram of rifampicin, so 3.5 times the current dose, and had it well tolerated. So they're planning to take that dose into a 2-month study, again in the standard regimen, beginning early next year, and we'll be excited to see those results. In addition, the Panacea group is currently doing a 2-month study with a rifampicin dose of 20 milligrams per kilogram, so twice the standard dose, and those results are expected to be available early next year. So again, we'll wait to see those results with interest. Next, I want to share with you some really intriguing and some data that Jerry Davies presented from the University of Liverpool and working with a group in Malawi. They performed a 200-patient study looking for a potential surrogate of long-term cure. So their study was set up to look at serial TTP measurements over time. So TTP uses the midget and is a measure of quantitative sputum bacterial load. So you follow the bacterial load going down, TTP actually goes up, but you follow the bacterial load going down over time. This has been done before, but what's really exciting is that these patients were then followed for long-term outcomes. The data set is 200 patients. The analyses presented were of only the first 130 patients. The full data set will be analyzed and available by the end of this year or very early next year. What the data showed was that the slope of the change in TTP over time correlated highly significantly with outcome, cure or failure, relapse at 30 months. The p-value was 0.0001. So this is really a very exciting preliminary finding. And one of the reasons I'm particularly excited about it and sharing it with you is that we really have the opportunity to build on that finding. It's a small data set, it's intriguing. We'll be able to work with Jerry when the full data set becomes available and formulate an exact hypothesis about what a surrogate endpoint out to 2 months that can predict long-term outcome is. We'll take that hypothesis. We have our REMOX phase 3 trial with 2,000 patients that will have database lock in the summer of next year, and we'll have results at the end of next year. Before we unblind, we'll be able to change the statistical analysis plan and take what was a retrospect, what was planned to be an exploratory analysis, because we have TTP over time built into that study. We'll take what was planned to be an exploratory analysis and turn it into a prospective test of this potential surrogate marker. So really by the end of next year, we could have a prospectively validated surrogate for long-term outcome in tuberculosis. That's one of the major goals of the CPTR organization. No promises, we need luck, the data need to come out right, but it's just a very exciting prospect to have on the horizon. I want to share with you a few other things going on in the field that many of you may not be aware of. Last November, a consultants meeting to look at optimal design and execution of phase 2 and phase 3 trials was held in London. That was last November. The sponsors of that meeting were CDC, NIH, TB Alliance, and CPTR. And a couple of action items came out of that meeting, and 2 of them are shown on this slide. They're projects. One was for the group to develop a universal mycobacteriology laboratory manual that could be used, that could be validated, made publicly available, and used in the future by future sponsors. Could either use it as a finished product or use it as a starting point to make the setup and standardization and monitoring of mycobacteriology in clinical trials easier. And the second was a similar approach to analyzing what I've called here EBA data. Really, we're talking about change in sputum bacterial load over time, whether it's 2 days, 2 weeks, or 2 months, but to create a standardized product with software available that, again, would be made publicly available. The process is to have a small group working on these and then open it to, again, a much larger group for final fine-tuning and validation. Both these projects are targeted to produce their outputs by next summer. Another project that many of you may have lost sight of is the off-the-tub gadifloxacin phase 3 trial. So this is a 2,000 patient trial looking at gadifloxacin substituted for ethambutol in the standard regimen, and looking at a treatment of 4 months versus the 6 months of the standard regimen to ask whether that can result in treatment shortening. This trial was conducted by the TDR arm of WHO and completed with 2-year follow-up at least a year ago, and then was sort of lost sight of. And I think many of you heard that there were issues with the database and difficulty with getting data into the database, out of the database. So I'm happy to report that those issues have been solved. There has been a transfer of the data in the database to a new database. Data that had not yet been entered are undergoing data entry, and the results of that phase 3 trial now are targeted to be available next summer. So I think very good news on that front. The final thing I want to leave you with is looking a little further into the future. And in order to look into the future, looking a little further back in the development path, and that's potential new regimens identified in Eric Nürnberger's mouse relapse model. This is a model that we think has, at least in the past, generally predicted treatment length in humans. And what you see here is that there now are a number of regimens with predicted treatment lengths of 6 weeks to 2 months. And you can see them there in the left column. Many of these, most of these regimens, all of the regimens in that column actually contain pyrazinamide. So those of you that participated in the TB diagnostics workshop the last couple of days will recognize why developing a rapid sensitivity to test to pyrazinamide is so high on our priority list. However, there are other new regimens that look very promising, shown right below that, consisting entirely of new chemical entities, or of new chemical entities and other entities to which there's no pre-existing molecular resistance, in particular clofazimine. So those data are shown below. I'll highlight one in particular, bedaquiline, PA24, and sutazolid. Entirely new chemical entities, looking like a potential 4-month regimen. And then above it, we still have the results pending, should have them before the end of the year. If we add Z to that, we expect to take that regimen down to the 6-week to 2-month category. So very exciting. In addition, another thing to point out, the regimen shown at the bottom is the first 2-month regimen we've identified that does not contain pyrazinamide. So again, very exciting. That's bedaquiline, a new nitramidazole, TBA 354, and sutazolid. So again, looking into the future, we're continuing to make progress. And with that, I'll close. Thank you.